Hello friends, this is Professor R.M. Mahendrakar from JSS Art, Science and Commerce College, Gokak. In a multicellular organisms, there are tissues, organs and organ systems. All the cells contain all the chromosomes and all the genes, but all the genes are not active in all the cells at all the time. Only some genes become active when their protein is required and other genes are switched off when they are not required. Hence, when the product of the gene is required, the gene is switched on, and when it is not required, it is switched off. This clearly explains that there is a gene regulation mechanism in all the cells of each and every organism. So in this video, I shall explain the gene regulation mechanism in prokaryotes and eukaryotes. The prokaryotic organisms are single-celled organisms that lack a cell nucleus, and their DNA therefore floats freely in the cell cytoplasm. The process of transcription and translation occurs almost simultaneously during protein synthesis. In prokaryotes, when protein is needed, transcription occurs, and when the resulting protein is no longer needed, transcription stops. So, the primary method to control what type of proteins and how much of each protein is expressed in a prokaryotic cell is the regulation of DNA transcription. All of the subsequent steps automatically follow. Thus, in prokaryotic cell, the control of gene expression is mostly at transcriptional level. An eukaryotic cell has evolved. The complexity of the control of gene expression also increased. For example, with the evolution of eukaryotic cell came the compartmentalization of important cellular components and cellular processes. A nuclear region that contains the DNA was formed. Transcription and translations were physically separated into two different cellular components. This therefore became possible to control gene expression by regulating transcription in the nucleus and also by controlling the RNA levels and protein translation present outside the nucleus. Some cellular processes arose to defend itself. Cellular processes such as gene silencing was developed to protect the cell from viral or parasitic infection. If the cell could quickly shut off gene expression for a short period of time, it would be able to survive an infection when other organisms could not. Therefore, organisms evolved a new process that helped it survive, and it was able to pass this new development to offsprings. Thus, gene regulation in eukaryotes take place at different levels during protein synthesis. Now, let us see at what all stages the regulation of gene expression is seen in a eukaryotic organism. First, regulation may occur when the DNA is uncoiled and loosened from nucleosomes to bind transcription factors. So this we call it as epigenetic level. Second is when the RNA is transcribed. This we call it as transcriptional level. Third is when the post-transcriptional modifications take place. That is, the primary transcript undergoes modifications to give rise to a matured RNA. So this level of regulation, we call it as post-transcriptional regulations. And then the fourth level is during the translation processes. And after the protein is formed, then the protein undergoes certain modifications. So this type of regulation, we call it as post-translation level. Thus, in eukaryotes, the gene regulation can take place at 5 levels. One is at the epigenetic level, Second is at the transcriptional level. Third is during the post-transcriptional level. Fourth is translation level and post-translational level. Now let us study about the gene regulation in prokaryotes. In prokaryotes, the structural genes of related functions are often organized together on the genome and transcribed together under the control of a single promoter. The operon regulatory regions include both promoter and operator. If a repressor binds to the promoter, then the structural gene will not be transcribed. Alternatively, activators may bind to the regulator region and enhance transcription. French scientist Francois Jacob and Jacques Monod at the Pasteur Institute were the first to show the organization of bacterial gene into operons through their studies on lac operon of E. coli. They found that in E. coli, all the structural genes that encode enzymes needed to use lactose as an energy source lie next to each other in lac operon under the control of a single promoter, the lac promoter. For this work, they won a Nobel Prize in Physiology in the year 1965. In prokaryotes, structural proteins with related functions are usually encoded together within the genome in a block called an operon and are transcribed together under the control of a single promoter. The operon includes promoter, operator, structural, and regulator genes. In prokaryotes, there are two types of operons. One type is that operon whose gene products are required consistently and whose expression therefore is unregulated. That is, these operons are never switched off. Such operons are constitutively expressed, meaning they are transcribed and translated continuously to provide the cell with constant intermediate levels of protein products. Such genes encode enzymes that are involved in housekeeping functions required for cellular maintenance, including DNA replication, repairs, and expression, as well as genes involved in core metabolism. Another type of operons are those who are expressed only when needed and are regulated by repressor, activators, and inducers. In this type of operon, genes are often controlled by extracellular signals. In case of bacteria, this typically means molecules present in the growth medium. These signals are communicated to the genes by regulatory proteins, which come in two types: positive regulators or activators and negative regulators or repressors. There are three types of regulatory molecules that can affect the expression of operons. They are repressor, activators, and inducers. Repressors are the proteins that support transcription of a gene in response to an external stimulus. In other words, a repressor keeps a gene off. Activators. Activators are the proteins that increase the transcription of an gene in response to an external stimuli. In other words, they accelerate the operon activity and switches the gene on. Inducers. Inducers are small molecules that either activate or repress transcription depending upon the need of the cell and the availability of the substrates. Inducers basically help to speed up or slow down, on or off, by binding to a repressor or activator. In other words, the inducers, they don't work alone. Now first let us study about the repressor type of operon of prokaryotes, taking the example of tryptophan operon in E. coli. E. coli has certain operons and other gene systems that enable it to manufacture any amino acid that is lacking in the medium in which it is placed, so that it can grow and reproduce. When an amino acid is present in the growth medium, the gene encoding the enzyme for biosynthetic pathway for that amino acids are turned off. And when the amino acid is not available in the medium they grow, the genes encoding the enzymes for the biosynthetic pathway for that amino acid are turned on. We call amino acid biosynthesis operon controlled in this way as repressible operons. Let us understand the mechanism, taking the example of the tryptophan operon. Like all operons, the tryptophan operon also consists of 5 structural genes: a regulator, promoter, operator, and terminator. Regulation of transcription occurs at initiation and termination site. The entire tryptophan operon is approximately 7,000 base pairs long. In E. coli, 5 structural tryptophan genes are present, namely tryptophan E, tryptophan D, tryptophan C, tryptophan B, and tryptophan A. They encode enzymes that synthesize the amino acid tryptophan. These genes are expressed efficiently only when tryptophan level is less in the medium. The promoter and the operator regions are upstream from the tryptophan E gene. There is a short region called tryptophan L, or the leader region. Within this leader region and close to the tryptophan E gene is a attenuator site that plays an important role in the regulation of the tryptophan operon. The regulatory gene for the tryptophan operon is the tryptophan R gene, located some distance from the operon at the upstream end. The product of tryptophan R gene is a aporepressor protein. Now here is a table showing the function of each of the tryptophan operon genes. Tryptophan R gene, it codes for the repressor. P is the site of promoter. O is the operator, which is the site of attachment of RNA polymerase. Tryptophan E, this gene codes for the enzyme anthranilate. Tryptophan D codes for phosphoribosyl anthranilate transferase. Tryptophan C gene codes for phosphoribosyl anthranilate isomerase. Tryptophan B codes for tryptophan beta synthetase, and tryptophan A codes for tryptophan alpha synthetase. Under severe tryptophan starvation, that is, when the tryptophan amino acid is less in the medium, the tryptophan genes are expressed maximally and transcribe all the structural genes, which in turn biosynthesize their specific enzymes and result in synthesis of tryptophan amino acid. Now in this slide you can see the proteins, that is the polypeptides formed from each of these tryptophan structural genes, and how these genes are helping in synthesis of the amino acid tryptophan. Under less severe starvation, that is when the medium contains a sufficient quantity of tryptophan, the tryptophan genes are expressed at less than maximal level. This is accomplished by a mechanism that controls the ratio of full-length transcript, that includes the 5 tryptophan structural genes, to a short 140 base pair transcript, which is called as the attenuator mRNA. The short transcripts are terminated by a process called as attenuation. The proportion of the transcript that includes the structural gene is inversely related to the amount of tryptophan in the cell. In other words, the more tryptophan present in the medium, the greater is the proportion of short transcript and lesser is the production of the maximal transcripts. Attenuation can reduce transcription of tryptophan operon. Thus, repression and attenuation together can regulate the transcription of the tryptophan operon. Let us see how this takes place. In a tryptophan operon, genes are controlled by a repressor. Here, tryptophan that controls the activity of the repressor acts as an co-repressor. That is, when tryptophan is present in the medium, it binds to the tryptophan repressor and induces a conformational change in that protein, enabling it to bind to the tryptophan operator and prevent transcription. Now here, this TRPR, that is the tryptophan regulator gene, it synthesizes this tryptophan repressor, and in the medium, when the tryptophan molecules are more in number, these tryptophan molecules bind with the repressor and brings about the change in its structure and enable this transformed structure to bind with the operator gene and thus prevent transcription. And here this attenuation brings in the reduction in the rate of transcription. So what is this attenuation? When tryptophan levels are high, RNA polymerase that has initiated a transcription pauses at a specific site and then terminates before getting to the tryptophan E gene. Attenuation and the way it overcomes rely on the close link between transcription and translation in bacteria and the ability of RNA to form alternative structures through intramolar base pairing. The key to understanding attenuation came from examining the sequences of the 5' end of the tryptophan operon mRNA. This analysis revealed that 161 nucleotides of RNA are made from tryptophan promoter before RNA polymerase encounters the first codon of TRPE. Near the end of the sequence and before the TRPE is a transcription terminator. It is composed of a characteristic hairpin loop in the RNA, followed by 8 uridine residues, and this so-called attenuator RNA synthesis usually stops, yielding a leader RNA 139 nucleotides long, and this mRNA, we call it as attenuated mRNA. Now this attenuated mRNA is produced only when the tryptophan level is high in the medium. But when in the medium the tryptophan amino acid concentration is low, so now the bacteria has to synthesize the tryptophan on its own. So when the tryptophan concentration lowers, the TRP repressor is free from its co-repressor and vacates its operator, allowing the synthesis of TRP mRNA and commence from the adjacent promoter. Now here the TRPR gene produces this TRP repressor. Now as in the medium tryptophan amino acid is less in number, the tryptophan amino acids cannot bind to the repressor and there is no conformational change in the repressor. So this repressor cannot bind with the operator, and when that repressor cannot bind with the operator, the RNA polymerase binds with the operator region and transcribe all the structural genes, and these structural genes that are transcribed undergo translation and produce all the 5 enzymes which are needed for the synthesis of the amino acid tryptophan. Thus, in this repressor operon, if the product is present in the medium, the operon is switched off. And if that product is not there in the medium, then the operon is switched on. So such type of operon we call it as repressor operon. Now we shall see the second type, that is the activator operon in E. coli. Just as the tryptophan operon is negatively regulated by tryptophan molecules, there are proteins that bind to the operator sequences and act as a positive regulator and accelerate the transcription process. To explain this mechanism, let me take the example of this lac operon of E. coli bacteria. First, let's study the structure of this lac operon. The lac operon consists of 3 structural genes: a regulator gene, a promoter gene, and a operator gene, and terminators. The 3 structural genes are LAC-Z, LAC-Y, and LAC-A. LAC-Z encodes beta-galactosidase enzyme, an intracellular enzyme that cleaves the disaccharide lactose into glucose and galactose and catalyzes isomerization of lactose to allolactose. The LAC-Y gene encodes beta-galactosidase permease. This is an inner membrane bound symporter that pumps the lactose into the cell during a proton gradient. The LAC-A gene encodes for the beta-galactosidase transacetylase, an enzyme that transfers a acetyl group from acetyl coenzyme A to beta-galactosidase. Only LAC-Z and LAC-Y are necessary for lactose catabolism. In E. coli, when glucose level drops, it has to look for alternative forms of sugar as a source of energy. So here, in this case, it is lactose. So when glucose level drops, the cyclic AMP molecules begin to accumulate in the cell. This cyclic CMP molecules is a signaling molecule that is involved in glucose and energy metabolism in E. coli. When glucose level declines in the cell, the accumulating cyclic AMP binds to the positive regulator molecule, namely catabolite activator protein, which is commonly called as the CAP protein. When cyclic AMP binds to the CAP, the complex then binds to the promoter region of the gene that are needed to use the alternate sugar, and thus the binding of the cap to the promoter increases the ability of the RNA polymerase to promoter regions, and the transcription of the genes and the protein synthesis is accelerated. Now, when there is sufficient quantity of sugar in the medium, there is no need for the alternative form of sugar. Hence, the cyclic AMP does not accumulate. So in absence of the cAMP, the cap cannot bind with the promoter, and hence the rate of the protein synthesis occurs, but in a slower rate. Thus, the activators are the molecules which enhance the rate of protein synthesis. So when this activating molecule binds with the promoter, the rate of protein synthesis increases. And when this activator doesn't bind to the promoter, the rate of the biosynthesis becomes very slow. The third type is the inducer operon in E. coli. In this type of operon, the substrate molecules act as an inducer molecule to switch on the operon. Let me explain, taking the classical example of lac operon. The expression of lac operon is regulated by lac repressor. The lac repressor has affinity to bind with the short DNA segment of the lac operator in absence of inducer and stop the process of transcription. So now let us see what happens when inducers are present, that is, when the medium contains a large quantity of lactose. Then the bacteria has to synthesize the lactose digesting enzymes, that is, the beta galactosidase enzyme has to be increased. Hence, when the lactose molecules are more, what happens we shall see. So lac repressor gene first synthesizes the repressor protein, and this lac repressor protein, if the inducers are present, that is when the medium is rich in lactose, here that lactose molecule act as an inducer, and this natural inducer of lactose is this allolactose, a metabolite of lactose. Now each subunit of repressor has one binding site for inducer. So these lac repressors, in presence of these inducers allolactose, they bind with this lac repressor and form a inducer repressor complex, and this inducer repressor complex alters the structure of the lac repressor, and because of this the lac repressor cannot go and bind with the lac operator. So now what happens is the RNA polymerase enzyme binds with the promoter and brings about the synthesis of the transcription of all these structural genes, and then these structural genes undergo the translation and produce the three enzymes which are responsible for the catabolism of this lactose sugar. Now, in absence of inducers, that is, when the bacteria has consumed all the lactose that is present in the medium, so now there is no need for the lactose catabolizing enzymes. Hence, the system has to switch off. So how it takes place? So this lac repressor R gene produces or synthesizes the lac repressors, and these lac repressors, because there are no inducers in the medium, the inducer repressor complex cannot be formed, so there is no change in the structure, and this lac repressor, which is having affinity to bind with the lac operator, binds with the lac operator gene and prevents the activity of the RNA polymerase enzyme. So the RNA polymerase enzyme cannot slide further, and hence the transcription of the structural genes LAC-Z, LAC-Y and LAC-A doesn't take place. In other words, the operon is switched off. Thus, in prokaryotes, the gene regulation takes place at transcription level only, and at transcription level there are three types of operons that are operating. One is the repressor operon, second is the activator operon, and third is the inducer operon. And all these are those operons which are switched on and off as and when needed. And there is another type of operon which never gets switched off. So such type of gene regulation occurs in prokaryotes. So thank you. Take care of yourself and register your name for vaccination through the Arogya Setu app. Once again, thank you.